Good afternoon, everyone. So we will discuss for today the fungus-like organisms of veterinary importance. So we have three organisms here, the Pythium insidiosum, Dinosporidium siberi, and Lacasia, formerly loboa. Then this is Lacasia loboi, formerly loboa loboi. So we'll start with the Pythium insidiosum, also known as Hypomyces destruens, belonging under the class Oomycetes, member of the kingdom Chromista. Can be found in the stagnant inland waters and occasionally in soil. The genus contains important plant pathogens. It grows on wide variety of media. Produce motile zoospores invading the animal tissue with mild abrasions. Causes cutaneous pityiosis in horses and gastrointestinal pityiosis in dogs. The usual habitat is the stagnant inland waters and occasionally in soil. Pathogenicity, motile zoospore apparently attracted by chemotaxis to wounds and abrasions on skin or intestinal mucosa. Insist on the exposed tissue. The encysted zoospore secrete a sticky material which is known to be possibly a glycoprotein that allows adhesion to tissues prior to invasion. Accepted hyphae develop from germ tubes produced by the zoospores at body temperature. It may extend into the tissues and may invade blood vessel, facilitating dissemination or spread and producing thrombosis. While in horses, pityiosis in horses, cutaneous pityiosis is the usual presentation in horses involving the skin. Intestinal or the enteric pityiosis also was reported. Lesions are found in the limbs that may occur due to contact with water-containing zoospores. Lesions are large, circular granulomatous nodules which often ulcerate. Sinus tracts may develop, exuding a serosanguineous discharge. Pruritus or intense itchiness is also marked. Necrotic yellowish coral-like masses, known also as cankers or leeches, can be removed intact from granulomas. Treatment is by surgical excision of lesions followed by immunotherapy. Enteric pityiosis is characterized by stenotic fibrous gastrointestinal lesions. Pityiosis in dogs involves the stomach and small intestine. Subcutaneous pityiosis is less commonly encountered. Clinical signs are vomiting, weight loss, intermittent diarrhea, and palpable abdominal masses. Extension of infection to pancreas, mesenteric lymph nodes, and bile ducts may occur. Cutaneous lesions, which are granulomatous with discharging sinus tracts, occur on the limbs, face, or tail. Treatment is by surgical excisions of lesions and by long-term treatment with antifungal drug itraconazole. Next, we have the Rhinosporidium siberi. It has not been cultured on inert media. It has been grown in monolayers of human rectal tumor cells found in stagnant water and soil. It belongs to the group of aquatic protista parasites class Mesomycetosea, kingdom Protozoa or Protista. It causes the condition rhinosporidiosis, a non-contagious pyogranulomatous lesions of the skin or mucosa. It was reported in dogs, horses, cattle, goats, and waterfowl, predominant in tropical and subtropical regions like Sri Lanka, southern India, and Argentina. For the pathogenicity and pathology, so the organism Rhinosporidium siberi is of low pathogenic potential and disseminated infection is rare. Presents most commonly as a chronic polypus rhinitis characterized by presence of large sporangia in affected tissues. Polyps may be sessile or pedunculated and up to 3 cm in diameter. Release of the endospores elicits a marked pyogranulomatous reaction. So it is the condition rhinosporidiosis is endemic in subtropical and tropical regions, also in North America and Europe. Infection occurs through minor trauma or in the skin or mucous membrane. Reddish-brown polyps in rhinosporidiosis may project from the nares and can occlude the nasal passages, resulting to noisy breathing exacerbated by exercise. Nasal discharge and epistaxis are also seen. Treatment is by cryosurgery or electrocauterization to avoid excessive bleeding, and diaminodiphenylsulfone or dapsone can be also used. Diagnostic procedure, nasal polyposis suggests the presence of the condition. Specimen for lab exam, like the biopsy material and scraping from lesions. Cytological examination demonstrate a neutrophilic response, and sporangia can be demonstrated in tissue sections. Okay, so in human, it causes chronic infection. In other animals, it is a chronic infection of the nasal and ocular mucosa, which includes in horses, humans, mules, cattle, dogs, goats, waterfowl. Direct examination from polyps produce sporangia or the white specks. Next, we have the Lacasia loboi or the Loboa loboi. Aquatic yeast-like organism has not been cultured in vitro, same with Rhinosporidium seborrhei. Causes lobomycosis or keloidal blastomycosis, which is a cutaneous lesions in humans and dolphins. Lobomycosis was not reported in domestic animals. Human cases were reported from tropical regions of South and Central America. Dolphins, like coast of Florida, result in skin changes range from white, crass to nodular or varicose lesion, which ulcerate easily and bleed. So last organism is the Pneumocystis carinii, a unicellular organism with a life cycle resembling a protozoal parasite. So Pneumocystis carinii was formerly classified under the protozoa, but now it is under the mycology na. And this one is also difficult to culture in vitro. Belong to the family Pneumocystidaceae, phylum Ascomycota. Trophic yeast and spore forms may be found in the lungs of affected animals. This is the cause of pneumonia among immunocompromised animals, dogs, and horses. Usual habitat, it is present in young, clinically normal humans and animals. So Pneumocystis carinii and Wakefieldiae are isolated from rats. Other Pneumocystis species are Girovicae, isolated from humans, Murina from laboratory mice, and Oryctolagi from old world rabbits. So this is known to be the cause of Pneumocystis pneumonia, or a small unicellular fungus that causes pneumonia. And it is the most prominent opportunistic infection among AIDS patients. This pneumonia forms secretions in the lungs that block breathing and can be rapidly fatal if not controlled with medication. Pentamidine and cotrimoxazole are the treatment of choice. So the usual habitat is present in young, clinically normal humans and animals. And for the transmission, the spread is via airborne. This is a suspected mode of transmission since it is the cause of pneumonia among immunocompromised individuals, especially among AIDS patients. In the animal body, the trophic form adheres in the cluster to type 1 pneumocytes, cause diffuse pulmonary consolidation, marked thickening of alveolar septa, and proteinaceous exudate in alveoli. For diagnostic procedures, specimen for lab exam includes lung tissue, bronchoalveolar lavage fluid. Cytological and histopathological specimen that are used for diagnosis are GEMS stain preparations and methanamine silver method stain, fluorescent conjugated monoclonal antibody techniques, immunocytochemical methods, electron microscopic examination of bronchoalveolar lavage fluid, and PCR. So pneumonia in dogs has been reported in horses, rabbits, cats, and pigs. Hereditary immunodeficiency is an explanation for the frequency of the disease in miniature Dachshunds, of breed of dogs. Arabian foals are also susceptible. Affected animals, seem to be afebrile, so without fever, and suffering from respiratory distress. TMPS, or trimethoprim sulfamethoxazole, administered for 2 weeks, is the drug of treatment or drug of choice. So that ends the fungus-like organisms. Thank you and have a good day.